Hello, my name is Joshua Mugabe. I welcome you to Proteins. What are proteins? How can protein diversity of living being be explained? Proteins are molecules that are made of sequences of amino acids bound by peptide bonds. The genetic code codifies 20 different amino acids that compose proteins. So there are numerous combinations of amino acids which can form polypeptide chains. And for this reason, protein molecules can be immensely diverse. What is the importance of proteins for living beings? Proteins play a fundamental role in nearly all biological processes. Due to their diversity, they can assume many different configurations, and they can play varied roles in cells and tissues. Some protein functions are noteworthy. The structure function, for example, the cell membrane proteins, cytoskeleton proteins, proteins of the connective tissues. There are others that function as the enzymatic functions. Enzymes are proteins in nature. Then others, the energy storage function. The proteins can be degraded into acetyl-CoA and cycle the Krebs cycle. The osmotic regulation function, for example, the albumin. Other proteins are for the transportation function, like the membrane channels, respiratory pigments. Others act as immune protection functions, for example, the antibodies. Then we have those ones, the proteins that are movement function, the contractile proteins. We have the endocrine integration functions. These are proteins like the hormones, and we have other proteins for the informative function like membrane receptors and intracellular signals. There are also many proteins whose biological functions are not yet known. What is the constitutional unit of proteins? The constitutional unit of proteins are the amino acids. What is an oligopeptide? How is it different from a polypeptide? The peptide is a molecule formed by the union of amino acids through the peptide bond. Oligopeptide is a peptide made of few amino acids. Oligo means few. Polypeptides, these are peptides with many amino acids. Poly means many, in general more than 50. How many unknown amino acids that form proteins in living beings? There are 20 different amino acids that form proteins related to the genetic code of living beings. There are still many other amino acids as yet not known. Does every amino acid have a central carbon? To which organic group is that central carbon bound? A carboxylic group, an amine group, and an atom of hydrogen and a variable radical R necessarily are bound to the central carbon of an amino acid. How can amine groups be classified? Amines can be classified into primary amines. These that which have only one R, the variable radical, attached to NH2. Secondary amines are those where one hydrogen of the NH2 is substituted by another R, thus having two R, which are variable radicals, and the tertiary amine are those with no hydrogen bound to the nitrogen, with three R's. What is the structural representation of a carboxylic group? Carboxyl groups have carbon attached to one hydroxyl group by a simple bond and to one oxygen by a double bond. The other site of binding in the carbon is available to other chemical entities. What is the structure flat representation of amino acid molecules? An amino acid has a central carbon to which the carboxyl group binds on a side and to which an R variable radical binds on the opposite side. In the perpendicular direction of those ligands, an amine group binds the central atom on one side and a hydrogen binds on the opposite side. The bind of carboxyl group to carbon, where a hydrogen is literally attached, is responsible for the name acid. In amino acids, and the bound of the amino group on the central carbon provides the name amino. What is the importance of the R group, variable radical, in an amino acid molecule? The R group is also called the lateral chain, is the variable part of the amino acid molecule. The R group can be a complex carbonic chain, a substituting methyl group, forming then amino acid alanine, or even only hydrogen, forming glycine, the simplest amino acid. So the R group is important because it is the differentiation factor of amino acids. How can binding of two amino acids for the peptide formation be described? A peptide is formed when a carbon from the carbonyl group of one amino acid is connected to the nitrogen of the amine group of another amino acid. During the binding, the hydroxyl of the carboxyl and one hydrogen of the amine is lost, resulting in the liberation of one water molecule. What is the binding between the two amino acids called? The chemical bond between two amino acids is called a peptide bond. Do the R group bound on the central carbons participate in the union between the amino acids? The peptide bond attaches the nitrogen of the amino group of one amino acid to the carbon of the carboxyl group of another amino acid, liberating one molecule of water. So the R group do not participate in that bond. Do the H group bound to the central carbons participate in the peptide bond. The central carbon themselves, the R group, and the hydrogen attached to the central carbons do not participate in the peptide bond. Do amine and the carboxyl group attached to the central carbons participate in the union between the amino acids? Yes. The nitrogen of the amine group of one amino acid binds to the carbon of the carboxyl group of another amino acid. The water molecule liberated from the formation of the peptide bond thus has hydrogen from the amine and an oxygen and another hydrogen from the carboxyl. Does the chemical reaction to unite amino acids incorporate or liberates atoms? What are the chemical entities incorporated or liberated in this reaction? The union of amino acids by peptide bond, it liberates atoms. They are liberated as constituent of one molecule of water. Are there different proteins made by the same total number of amino acids? Different proteins with the same total number of amino acids may exist. In such case, the differentiation is given by the type of amino acids and by the sequence in which they form the protein. Are proteins with the same number of each different amino acid that form them necessarily identical proteins? Even if many proteins have the same number of each different amino acids that form them, for example, 50 alanine, 70 glycine, and 20 histidines, the sequence in which these amino acids are connected may be very different. So, if two or more proteins are in such condition of numeric similarity for each type of their constituent amino acid, they are not necessarily identical. What is the essential condition for a protein to be identical to another protein? For a protein to be identical to another protein, it is necessary for the sequence of the amino acid that form them to be identical. What is the primary structure of a protein? What is the importance of the primary structure? The primary protein structure is the linear sequence of amino acids that form the molecule. Primary structure is the basis of the protein identity. Modification of one amino acid of the primary structure creates a different protein. Different proteins can be inactive or can even have other biological functions. What is the secondary structure of a protein? The secondary protein structure is generated by the manner its amino acids interact through the intermolecular bonds. These interactions create a spatial conformation of the polypeptide filament. The two most studied secondary conformation of proteins are the alpha helix and the beta sheets. What is the difference between the alpha helix and the beta sheet protein conformations? The alpha helix and beta sheet conformation are the two main types of secondary structure of a protein molecule. According to the primary protein structure, its secondary structure can be of one type or the other. In the alpha helix structure, the polypeptide curves longitudinally by the action of hydrogen bonds, forming a spiral or helix. In the beta sheet conformation, the protein is more distended and the hydrogen bond form a zigzag-shaped protein structure called beta strand. Many assembled beta strands make a beta sheet. What is the tertiary structure of a protein? What are the main types of the tertiary structure? The tertiary protein structure is a spatial conformation addition to the secondary structure, which the alpha helix or the beta sheet fold itself up. The forces that keep the tertiary structure generally are interaction between the R groups of the amino acids and between the other parts of the protein and water molecules of the solution. The main types of tertiary structures of protein are the globular proteins and the fibrous proteins. What is the quaternary structure of a protein? Do all proteins have quaternary structure? The quaternary protein structure is a spatial conformation due to interaction among polypeptide chains that form the protein. Only those proteins made up of 2 or more polypeptide chains have quaternary structure. Insulin has 2 chains. Hemoglobin has 4 chains. Immunoglobulin, the antibodies, they have 4 chains. Are some examples of proteins having quaternary structures. What is protein denaturation? Is there any change in the primary structure when a protein is denatured? Secondary, tertiary, and quaternary structure of protein are spatial structures. Denaturation is the modification of any of these spatial structures that makes a protein deficient or biologically inactive. After denaturation, the primary protein structure is not affected. How can denaturation be classified regarding to reversibility? Protein denaturation can be reversible or an irreversible process. That is, it may be possible or impossible to make a protein regain its original spatial conformation. What are some factors that can lead to protein denaturation? Protein denaturation can be caused by temperature variation, pH changes, change in the concentration of the surrounding solute, and other processes. Most protein denaturation after certain elevation of temperature or when in very acid or very basic solution. This one of the main reason that it is necessary for the organism to keep stable temperature and pH. Is it expected that the change in the primary and in the secondary or in the tertiary structure of a protein will produce more functional consequences? any change in the protein structure is irrelevant if it alters its biological activity. Changes in primary protein structure are more important because they are modification in the composition of the molecule, and such composition determines the other structure of the protein. In sickle cell anemia, a hereditary disease, there is substitution of one amino acid by another in the 4-polypeptide chain of hemoglobin. In this case, are all of the structure level of the protein modified? In sickle cell disease, there is a change in the primary protein structure of one polypeptide chain that forms hemoglobin. The amino acid glutamic acid is substituted by the amino acid valine in the beta chain. The spatial conformation of the molecule, in addition, is also affected and modified by this primary mistake, and a modification also creates different shape of the red blood cell, which is sickle. Modified sickled red blood cells sometimes aggregate and obstruct the peripheral circulation, causing tissue hypoxia and pain crisis typical of sickle cell anemia. What is the difference between essential and natural amino acids? Essential amino acids are those that the organism is not able to synthesize and that need to be ingested by the individual. Natural amino acids are those that are produced by the organism. Living being species that produce every amino acid they need. For example, bacteria, Escherichia coli, does not have essential amino acid. Other species like human need to obtain essential amino acid from the diet. Among the 20 different known amino acids that form protein, humans can make 12 of them, and the remaining 8 need to be taken from the proteins they ingested with birds. Food. The essential amino acids for humans are phenylalanine, histidine, isoleucine, lysine, methionine, threonine, tryptophan, and valine. What are the respective some remarkable functions of myosin? CD4, albumin, keratin, immunoglobulin, reverse transcriptase, hemoglobin, and insulin. Myosin is a protein that is associated with actin, and they produce muscular contraction. CD4 is a membrane protein of some lymphocytes, the cells that are infected by HIV. Albumin is an energy storage protein and also an important regulator of blood osmolarity. Keratin is a protein with a structure function present in the epidermis, skin appendages of vertebrae. Immunoglobulin, they are antibodies, specific proteins that attack and inactivate strange agents that enter the body. Reverse transcriptase is the enzyme responsible for transcription of RNA and formation of DNA in the life cycle of retroviruses. Hemoglobin is a protein that carries oxygen from the lungs to the cells. Insulin is a hormone secreted by the pancreas that participates in the metabolism of glucose. Thank you very much for participating in proteins. Please do like, share, and subscribe.